Hello students, welcome everyone. As you people know that in the last video we have studied part 1, that is structure of the flower of the chapter sexual reproduction in organism, which is under the unit reproduction. Now we'll move on to part 2, that is pre-fertilization structure and events, means the events which are occurring before the fertilization or prior to the fertilization, and what are all the structures involved in this particular pre-fertilization process. And in this part we'll be studying in detail about the andrecium, and we'll also study the structure of the stamen, microsporangium and the pollen grain. Now let us start with this. And what is andrecium? Andrecium is a layer of stamen or a whorl of stamen. It is the male reproductive part of the flower. It consists of a whorl of stamens or a layer of stamens. The number and the length of the stamens is different in different species. So as you can see in the diagram, what is the structure of the stamen? It is generally having two parts. One is filament and another one is anther. So what is filament? Filament is a long and slender stalk. It is attached to the thalamus or the petal of the flower at its one end. What is thalamus? Thalamus is also called as receptacle. It is the swollen part of the pedicel of the flower. What is pedicel? It is the stalk of the flower. Next part of the stamen is the anther. What is anther? It is terminal and typically bilobed in structure. That means having two lobes, and each lobe is having two thecae. So each lobe has two thecae, hence it is called as dithecous. So often a longitudinal groove runs lengthwise separating the theca. So there you can see a demarcation between the two thecae, and that can be seen as a proper demarcation in the anther. Now we'll move on to the transverse section of the anther. So we'll see the anther transverse section and enlarged view of the single microsporangium, which is showing a different layers of the microsporangium. So what is this? What you can see in the transverse section of the anther? The anther is generally a tetragonal structure, that means having four angles consisting of four microsporangia. These four microsporangia are located at the four corners of the anther. So each lobe consists of two microsporangia. The microsporangia develop inside the pollen sacs. Pollen sacs are the bag-like structures inside which the microsporangia will develop. So pollen sacs extend longitudinally, that means all through the length of an anther, and are packed within the pollen grains. That is, inside the pollen sacs, pollen grains are present. Next, what are the structure of the microsporangium and what are all the different layers of the microsporangium? As you can see in the diagram, there is an enlarged view of one microsporangium showing all the wall layers. So a typical microsporangium is roughly circular or round in outline. It is surrounded by four wall layers. So the outer epidermis, next comes the endothecium, next the middle layers, which are usually two in number, and the inner side comes the tapetum. So that outer three layers, so that is epidermis, endothecium and the middle layers, give protection and help in dehiscence of the anther. What is dehiscence? Opening of the anther. So why this opening of the anther will happen? To release the pollens which are present inside the pollen sacs. So what about the tapetum? The tapetum is the innermost layer which nourishes the developing pollen grains. Cells of the tapetum contains a thick or a dense cytoplasm and generally have more than one nucleus, that is multinucleate in structure. When the anther is young, a group of compactly arranged homogeneous, that means similar type of cells or parenchymatous cells called as sporogenous tissue occupies the center of each microsporangium. This is the structure of the microsporangium in an enlarged view. Now coming to the process or the event which is happening in the pre-fertilization, that is microsporogenesis. What is microsporogenesis? Microsporogenesis is divided into two words, microspore and genesis. That means genesis means origin or formation, which means origin or formation of the microspore. So we'll see what is microsporogenesis in detail. As the anther develops, each cell of sporogenous tissue undergo meiotic division. So you have studied mitotic and meiotic division in your first POC classes. So it undergoes a process of meiosis to form microspore tetrads. That means microspores are arranged in a cluster of 4 cells. So these are called as tetrads. Each one is a potential pollen. That means it is called as microspore mother cell. The potential pollen here is being called as microspore mother cell. Now, the formation of microspore from a pollen mother cell, that is PMC, through a process of meiosis is called as microsporogenesis. So it is, I'll repeat it once again, the formation of the microspores from a pollen mother cell through meiosis is called as microsporogenesis. As the anthers mature and dehydrate, that means as the water is lost in the anthers, the microspores separate from each other and develop into pollen grains. So each microsporangium now contains thousands of the pollen grains inside. So the pollen grains are released by the opening of the dehiscence or breaking of the anther. You can see the structure of a mature dehisced anther. That means the anther which is broken, from inside that you can see the pollen grains. Now we'll see the structure of the pollen grain in detail. So pollen grain is a male gametophyte. It is actually the male gametophyte. So generally it is spherical in structure, round in structure. And what is the diameter of a pollen grain? It is 25 to 50 micrometer. So the cytoplasm is present in it and it is surrounded by the outer covering or the outer layer called as plasma membrane, as every cell is having. Next, what is a pollen grain? A pollen grain, what is the structure of a pollen grain? It has 2 layers, outer exine and inner intine. What is exine? As you can see the diagram of a pollen grain, and you can see some of the pictures under the scanning electron microscope. You can see some of the pollen grains and you can make out the structure, variation in the structure and the shapes of the pollen grains. So we'll see what is exine and intine here. You can see the structure of the mature pollen grain in an enlarged view in the diagram. Exine, it is the hard outer layer made up of a highly resistant organic material called as sporopollenin. What is sporopollenin? It is resistant to high temperature, some of the strong acids and bases. So the enzymes cannot degrade or destroy or decompose the sporopollenin. Hence, exine can be preserved as fossil due to the presence of the sporopollenin. Exine exhibits different patterns and designs in different species of the plants. So exine is having a small aperture called as germ pores where the sporopollenin is absent. Except these germ pores, it is present everywhere on the exine layer. Next, coming to the inner layer, that is intine, it is inner wall. It is thin and continuous layer made up of 2 polysaccharides called as cellulose and pectin. Polysaccharides, these are the polymers of the carbohydrates. So a matured pollen grain contains 2 cells. So you can also see the stages of a microspore maturing into a pollen grain. Under that you can see the matured pollen grain and what are all the cells present in it in detail. It is having 2 cells. One is vegetative cell and generative cell. Vegetative cell, it is bigger in size and has abundant food reserve and a large irregularly shaped nucleus is also present. So what is generative cell? It is a small cell and it floats in the cytoplasm of the vegetative cell. It is spindle shaped. What is spindle shaped? It is in the shape of a fish. How the fish will be there? In the same shape it will be there. So with a thick or the dense cytoplasm and a nucleus. Next, coming to the next point, that is, in over 60% of the angiosperms, pollen grains are shed at the 2-cell stage. In some of the other plants, the generative cell divides mitotically, that is, the small cell divides mitotically to give rise to 2 male gametes before the pollen grains are shed. So, what happened to these shed pollen grains, which are shed from the stamens after the dehiscence of the anther? The shed pollen grains have to land on the stigma of the flower, same flower or different flower, before they lose the viability. What is this viability? It is the active state or the ability to work. So the viability period of the pollen grains is different in different species, and the viability also depends on temperature as well as humidity. So next we'll see some of the economic importance of the pollen grains. So pollen grains of some of the plants, so these are very rich in the nutrients, and these are used in some of the Western countries. Pollens are used in the form of tablets and syrups and used as a food supplement. Pollen tablets and syrups increases the performance of the athletes and the horses which are used in the racing. Pollen grains of some plants such as carrot grass and parthenium are allergic for some people. It also leads to chronic respiratory disorders like asthma and bronchitis, etc. Now, pollen grains can be stored for years in liquid nitrogen. That is, what is the temperature of the liquid nitrogen? Minus 186 degrees centigrade. So they are also used as pollen banks, similar to the seed banks. So pollen banks, what are the pollen banks? Here, the pollens of different plants or the trees are stored for many years and can be utilized for the process of breeding or the reproduction in different times. It is used in crop breeding programs. So these are some of the economic importance of the pollen grains. With this, I'll stop today's class. So let us recap what and all we have studied today. So we have studied the structure of the andrecium, in that the stamen, what is the stamen made up of? That is anther and the filament. Then we have studied the transverse section of the anther and an enlarged view of the microsporangium and what are all the different layers of the microsporangium. Next, we have seen some of the scanning electron micrographs of the pollen grains, and then enlarged view of the pollen grains. What is the structure and what are the different walls which are present? Next, we have seen the different viability of the different pollen grains, and then we have gone for the economic importance of the pollen grains. So thank you everyone for watching.